Today we're going to talk about epigenetics. The word epigenetics literally means on top of or in addition to genetics. So we're talking about phenotype changes that do not involve alterations to the actual DNA sequence. A couple of definitions that I pulled from Wikipedia include functionally relevant changes to the genome that do not involve a change in the nucleotide sequence, and the study of mitotically or meiotically heritable changes in gene function that cannot be explained by changes in the DNA sequence. What you see is sometimes these definitions include requirements for these to be heritable, and sometimes you don't. That's really not the most important part about this definition. What we're going to go over here is what these changes look like at the molecular level and how these might be influenced by external forces. The term epigenetics and the idea that these phenotypes can change without changing the DNA sequence itself goes back to 1942 and some work done by a guy named Conrad Waddington. He was working on fruit flies and noticed that when he accidentally left flies next to a heat source, let's say a light bulb, that the next generation of flies would have altered wing phenotypes, so different shaped wings, even when those progeny were raised away from the heat source, meaning that something was inherited that changed that phenotype, but this was unlikely to be a DNA mutation itself. And what he saw over time is that generationally, those phenotypes would shift back towards the wild type over time in the absence of any sort of other external forces. There are three main categories of epigenetic changes that could affect phenotype. Number one is DNA methylation. Number two is histone modification. And number three is noncoding RNA. And there are many different types of noncoding RNA. All three of these types of epigenetic changes can affect the way genes express and thus affect phenotype. These are heritable and also reversible over time. Let's focus first on DNA methylation. These types of epigenetic modifications occur at the promoter level. The promoter, you'll remember, is the region of DNA regulatory elements that are just upstream of any transcribed gene. These DNA regulatory elements are where certain proteins that are involved in transcriptional initiation will bind in a way that influences whether or not transcription begins and at what rate. Another common feature of any highly regulated eukaryotic gene promoter is the presence of CpG islands. These are very common in mammalian genomes. CpG stands for any cytosine that comes before a guanine in a 5' to 3' direction. The P in CpG island refers to the phosphate that lies in between those two sugars. CpG islands are rich in these CG combos, and you can see that on the opposite strand, CGs run backwards in the 5' to 3' direction on the opposite strand. These islands may be hundreds or even thousands of bases long, and in mammals at least, 70 to 80% of the cytosines in these CpG combos are methylated. Specialized enzymes that control whether or not methyl groups are added or take away regulate the presence or absence of methyl groups at these CpG islands. The presence or absence of methyl groups on these cytosines will dictate whether or not proteins interact with that region, and thus DNA methylation at these CpG islands is a major factor in whether or not a neighboring gene is transcribed at all. Now that we understand how DNA methylation works at the molecular level, let's take a look at how this form of transcriptional regulation might influence the phenotype of two different cells in an animal like a mosquito. We know from earlier chapters that every cell in this mosquito's body, or essentially every cell, has an identical copy of a genome. But The phenotypes of each cell vary because not every cell uses the genome in the exact same way. In some cells, a certain suite of genes are expressed, and in other cells, a different suite of genes is expressed. This is how we create unique phenotypes amongst all of our body cells using a common genome. Many of the olfactory cells or neurons of the antenna of the mosquito must express olfactory receptor 7 to be functional. However, those same cells on the antenna do not need to express the messenger RNA and therefore protein for channel rhodopsin beta, which is an important light-sensitive protein. Light-sensitive cells, however, in the mosquito eye must express that channel rhodopsin beta and do not need to express olfactory receptor 7 for their normal function. The primary way that two different cell types within a given organism regulate gene expression in a differential manner is through transcription factors. Each cell type in the animal expresses a unique suite of transcription factor proteins. In the olfactory cell at the top, OR activators are expressed to turn on the expression of the necessary odor receptors, olfactory receptors for function, and at the same time, channel rhodopsin repressors are expressed to make sure that none of the channel rhodopsin beta protein is made. Likewise, light-sensitive cells at the bottom express a totally unique set of transcription factors. In this case, the channel rhodopsin activator protein binds to the DNA regulatory sequence upstream of the channel rhodopsin, thus helping express more of it, and an OR repressor protein binds upstream of the olfactory receptor 7 gene and others like it to ensure that the expression of those receptors does not occur in light-sensitive cells where they would be useless and a waste of energy. DNA methylation as a form of epigenetic regulation has evolved in many eukaryotes as an extra way to regulate expression when perhaps transcription factor regulation isn't up to the task. For example, there may be some olfactory cells in which you do not want expression of olfactory receptor 7 for some particular reason. Perhaps those cells are sensitive to other odors requiring other receptors. In this case, it may be problematic to have an OR activator protein in that cell that is needed for the expression of some olfactory receptors, but not all. Here comes DNA methylation to solve the problem. Through the activity of methyltransferases, the promoter region just upstream of olfactory receptor 7 in olfactory cell A is modified. The cytosines on the CPG islands in this location have extra methyl groups added to them, thus changing their chemical identity. With their identity changed, OR activator proteins do not interact with that promoter in the same way they did before. Thus, transcription has been controlled at the DNA methylation level. Olfactory receptor 7 is not expressed in olfactory cell A, but it remains expressed in olfactory cell B. We have not changed the nucleotide sequence itself, we have only modified the DNA bases that were already there. Now that we've covered DNA methylation as a mechanism for epigenetic change that can influence gene expression and thus phenotype, let's look at how histone modification accomplishes basically the same thing mechanistically. Here we can see the double-stranded DNA that comprises a chromosome interacting with individual proteins called histones. Zoomed in here, we can see a single histone that has acetyl groups here, methyl groups, and phosphate groups attached to these little outcroppings from the protein that are referred to as histone tails. You can see that individual histones here in light blue are organized into nucleosomes, these little balls of proteins, and that DNA is wrapped around that about 2.5 times. These become larger and larger structures with organization, which become chromatin fibers. And as these continue to fold up in more complicated structures, we get essentially what we see on a microscope as a chromosome as it appears leading up to cell division. each nucleosome is organized like this. Okay, so there's 8 core units of that nucleosome represented by 2 each of H2A, H2B, H3, and H4 histones. And then the H1 histone serves a special purpose in that it serves as a linker between 2 neighboring nucleosome structures. Histones are programmed to interact with DNA specifically based on which modifications are present or absent on these individual histone tails. The chemical identity of these groups changes the relative charge of the histone and how this is going to interact with the negatively charged DNA. Let's look at one specific example of a histone modification. In this case, acetylation, so the addition of an acetyl group to a histone tail. You can see that regions of chromatin that are heavily acetylated at the top have a more open or relaxed configuration where the DNA is accessible to other proteins. The sequence is available. This would promote things like gene expression at those locations because their promoters would be out in the open and able to interact. On the bottom, if we take those acetyl groups away from the histones, you can see that we've encouraged a closed configuration, which will actually limit the amount of gene expression happening at this region. Acetyl groups are essentially neutralizers of the normally positive charged histone tails. And that's important because the positive charge of the histones is what promotes tight conformation between the DNA and the histone itself. The DNA, of course, is negatively charged thanks to the phosphates on the backbone there. So when we add acetyl groups, we're limiting that positive charge on the histones, and thus DNA is less attracted to the histone itself. Whereas if we take those acetyl groups away, the positively charged histones are more tightly interacting with the negatively charged DNA. The third type of epigenetic modification is any influence from non-coding RNAs which affects gene expression. Here I have a very simple diagram of how this mechanism works. MicroRNAs are transcribed just like coding genes are, only they do not contain open reading frames. They don't need to become proteins to have function. You can see that microRNAs are often synthesized as first longer pre-microRNAs. A number of proteins then process these into smaller pieces, and along with a dicer complex of proteins outside of the nucleus, these microRNAs act as guides for these proteins, much in the same way that guide RNAs guide CRISPR-Cas proteins to cut. MicroRNAs guide something called a RISC complex. You don't really have to know what that is, but a specialized set of proteins that will go recognize a complementary messenger RNA that's out here. This messenger RNA normally would be being translated by the ribosome here and turning into a protein. But if the presence of this microRNA in this particular cell is there and is guiding these RISC complexes, 2 things can happen. One, the mRNA itself can be degraded from that activity of the microRNA, or simply the microRNA could block translation, keeping those ribosomes away. In either case, you're turning down the gene expression of that target messenger RNA in a way that's probably going to affect phenotype. So in this way, we are affecting gene expression and phenotype without directly changing the DNA sequence that would be here in the nucleus. We're not affecting the genome itself. So this is why non-coding RNAs are considered sort of the third pillar of epigenetic changes. I think that provides you students with a nice overview of how DNA methylation, histone modifications, and non-coding RNAs can affect gene expression without changing the DNA sequence itself. The reason why all of these things are important is because environmental factors, take for example, a healthy, well-rounded diet. This could apply to things like not smoking cigarettes or avoiding certain chemical contamination in our foods. These types of environmental factors affect things like mitochondrial function. They affect other processes within our bodies. And the way that manifests is those directly affect the activity of some of the proteins we've talked about, histone acetyltransferases or deacetylases, DNA methyltransferases, all the readers, writers, and erasers that modify our histone tails, that control how much and where methylation occurs on our chromosomes. These affect directly the activity of non-coding RNAs. All of these will affect transcription and translation and thus change phenotype. So we literally are what we eat. We are a product of not only our genetic code, which is genetics, but because of these environmental factors, our epigenetic factors are changing and affecting our phenotype. This, for example, is why identical twins oftentimes have somewhat different or even very different phenotypes, not just in how they look, but how they might behave or the things that they may feel. And that's because the identical twins are not experiencing the exact same environmental influences throughout their lives and throughout development. And this is going to affect their phenotypes in a different way. And remember, too, that these epigenetic changes are heritable. And so the diets of parents can affect the gene expression and thus phenotype of their children and their children's children. But they're also reversible, meaning if those environmental factors are more positive for later generations, that these methylation patterns on the DNA and the chemical modifications to histones will revert back to a maybe more stably functional type, or the behavior of non-coding RNAs may, inside the cell, may change back to a more beneficial phenotype. Well, I hope you've learned a lot today about what epigenetics is and how it affects living things. The key here is to remember that every genome in every cell of your body or any organism's body is identical, yet how each of those cells express those genes is really based on epigenetic factors influencing gene expression. Some of them are by design. For example, in the mosquito, the difference between an olfactory cell and a light-sensitive cell may be completely dictated by epigenetic differences using the same genome. And it can also change the way cells function over time. On the picture here on the left, this man is living a very healthy lifestyle with exercise and good diet. The genome that he was born with expresses genes in a way that lead to sort of an overall healthy phenotype, where over here on the right, the lack of exercise and a somewhat unhealthy diet lead to a completely different phenotype. And in both situations, the same genome is at the heart of those cells. So just something to think about. I hope you enjoyed the video.